Hi, everybody. Thank you for joining us in the Synthetic Intelligence Forum. My name is Vic, and I'm the founder and the host of this forum. Today, I'm very happy to be welcoming back to the forum a dear friend, James Zou. Professor Zou is an associate professor in the Department of Biomedical Data Science at Stanford University, and he also holds courtesy appointments in the departments of computer science and electrical engineering. I'm really excited that Professor Zou is going to talk to us today about the applications of generative multi-agent systems for advancing scientific research. So welcome back to the forum, James. It's a pleasure for us to have you back with our community of practitioners and researchers of data science and artificial intelligence, and very excited to hear from you about virtual labs. So the floor is yours. Please take us away.
Well, thank you very much, Vic. It's always a pleasure to come back and to have these discussions with you. Yeah, so excited to share some of the recent projects we're doing around AI scientists. And the setup for this is that, usually when we think about AI, right, we're thinking of AI as a tool for solving some specific task, right? For example, AlphaFold is a very useful tool for biology and for medicine for predicting the structures of different proteins. And some of the other work that we've done before, like EchoNet, is also a very useful tool for assessing these cardiovascular ultrasound videos, right? When we think of AI as a tool, then it's really used as a very specific part of a much larger workflow, right? In scientific applications, then usually the scientists have to figure out what data to feed into these tools, and also spend a lot of time thinking about how to interpret the data that come out of these tools. Right? So it's actually still a lot of the work that goes beyond using the specific tool. And I think we're really in this interesting paradigm shift now, especially with the breakthroughs in these large language models and agents, where I think we can start to think about AI as a partner in the scientific research rather than just simply as a tool for solving a specific task. Right? For example, now with these language models, now we can actually have a language model that uses tools like AlphaFold. We can also have a team of language models that communicate with each other, right, through natural language, and also communicate with different databases and with human researchers. So I'm really excited about this potential, right, of thinking about AI scientists as a partner that goes beyond a specific tool. And to help us to realize this vision as AI as a partner, recently with my students, we developed this platform that we call the Virtual Lab. So the idea of the Virtual Lab is that it's a team of interdisciplinary AI scientist agents. It's led by actually an AI professor, and under the AI professor, there's sort of like a team of different AI students with different expertise, like in computer science, in machine learning or biology. And the idea is that this team of virtual agents, virtual scientists, would actually work closely with each other, and they will also collaborate with the human researchers to try to tackle quite challenging and open-ended research projects. And just to make this very concrete, I want to give an illustrative example of how we worked with the virtual lab and how the virtual lab actually designed new binders, right, to the recent SARS-CoV variants. And this virtual lab is developed by Kyle Swanson, who's a fantastic PhD student working with me at Stanford. So here's sort of an overview of the virtual lab, right? So we set it up to be very flexible, right? So when individual human researchers, the way that we interact with the virtual lab is primarily by interacting and talking with the professor agent, this PI agent, right? So maybe we'll tell this PI agent in this case that we are interested in designing some potential therapeutics or vaccines for the recent COVID variants. And then the professor agent then would actually automatically create the different sub-agents that have the relevant expertise, right? So for this problem, maybe the professor agent will create like an immunologist, a computational biology agent, and a machine learning specialist. But for other problems, the professor agent, the PI, could actually create different agents that have the relevant expertise. And this is basically the job of the professor agent then to create and also to train and to develop each of these sub-agents with their relevant expertise. And the way that's basically under the hood of each of these agents is a GPT-4o model. And the way that these agents develop the relevant expertise is really by using often the sophisticated tools in the relevant domains. Right? So for example, the computational biologist needs to learn to use like AlphaFold and other sophisticated tools. So once the virtual lab is created, then they start to make progress towards solving some quite challenging and open-ended research problems through two types of meetings, right? So they have group meetings where all the agents discuss together, and then one-on-one meetings, right, where a specific agent tries to solve a specific task. So here's a bit more information about these meeting structures. right? So we designed the group meetings to be one where all of the agents actually sort of contribute and discuss together, right? So in a group meeting, the professor agent would say, okay, here's the meeting agenda. Maybe here are some questions that they want to answer by the end of this meeting. And then it will share this agenda with all of the participants. And then all of the participating agents then will go around sort of in sort of like a virtual table to provide their feedback and updates that relates to those meeting agenda. Right, so here's an example of one of these virtual lab group meetings. This is actually one of the earlier meetings where the PI, the professor agent, first says that, okay, so here's the overall context and agenda, right? The goal of this meeting, the goal of this project is that they want to develop some sort of binders that can bind to the recent variants of the SARS-CoV-2 spike protein, right? And the reason why that's important is because for the recent variants of the virus, the existing vaccines are not as effective, right? And then the agent starts to contribute ideas, right? So first you see the immunologist actually contributed quite an interesting idea. It suggests that we should actually design some sort of nanobodies. And for the context here is that nanobodies, you can think of them as sort of like much smaller versions of the regular antibodies, right? Most of the time, often people would think about using antibodies, which are more widely available from different animals. And nanobodies is only available in very few animals like camels. But the immunologist said that, okay, even though it's a bit unusual, but it's actually good to design nanobodies rather than antibodies, right? Because these nanobodies are smaller, so easier to model. And then the machine learning agent actually chimed in and says, okay, I agree with focusing on nanobodies, right? They're smaller, which simplifies the computational task, and also suggests using deep learning models to predict the structures of these nanobodies. And then the computational biologist also provided a useful comment, which is that, well, in addition to using the deep learning models, let's also use some more like physics-style molecular dynamics and docking, right, to also make it more robust. And for every meeting, we also have like a scientific critic agent, which is, you can think of this as sort of like a reviewer agent whose job is to actually provide some constructive feedback and criticisms, right, around the decisions and ideas proposed by the other agents. Right. In this case, the critic agent actually says that, well, there's some potential issues we want to be aware of when using these deep learning models for prediction, especially because there's relatively limited amount of data for these nanobodies, right? So there are potential biases and we don't want to overfit. So this is sort of like a snippet of one of these meetings, but the actual meetings can actually go on for multiple iterations and typically it's much longer. So that's the group meeting. And then in an individual meeting, a one-on-one meeting, the goal of that is basically for an individual agent, right, to solve now like a specific subtask, right? So maybe one subtask could be like writing a particular piece of code. Then that individual agent will meet with maybe the critic or with the PI agent and try to solve that problem. So one of the nice things about these virtual scientists is that their meetings are much more efficient than our meetings. So one of these virtual lab meetings maybe will take a minute or a couple of minutes, sometimes less. So in the time that we had our coffee, they already had potentially hundreds of meetings. And moreover, these virtual agents, they don't get tired, right? So they can actually run multiple meetings in parallel. So what we actually do is that basically for every decision that's made by the virtual lab, we run 5 meetings in parallel, discussing the same questions, the same agenda. And different times when you discuss the same question, sometimes the virtual lab agents will actually come up with different ideas, right? Different decisions. So this is actually quite a useful way to use these parallel meetings to explore the different possibilities. And after these parallel meetings, then the professor agent will look at all the summaries of these meetings and make a kind of a consensus decision, right? And then discuss the next steps. And in case you're curious, so here's basically a breakdown of how often do different participants participate and speak, right, in the virtual lab. So the human researchers, we don't speak very much, maybe about 1% of the time, because our job is to really provide more like these high-level feedbacks, right? So the principal investigator, the PI agent, actually speaks quite a bit, about 20% of the time. The person that speaks the most is the computational biologist agent, right? And I think this makes sense here because given the goal is to design these binders to COVID, right, the computational biology agent actually has the most work to do. And you can also see that the scientific critic, this reviewer agent, also speaks quite a bit because it has to provide some feedback to every decision that's made by the virtual lab. And all of the work here by the hands-on work is all done by the AI agents, right? So for example, all of the code that's used by the virtual lab is all written by the computer scientist agent. So we don't write any code for the agents. They do their own work. And moreover, these agents really come up with their own ideas, right? Saw that they actually decided to design nanobodies, which is sort of already a surprising choice. And once they made that decision, they also have to come up with their own workflow for how to create or how to optimize these nanobodies. And this is where I think the virtual lab agents actually made some quite interesting and in my view also somewhat innovative designs. So first they created this computational workflow that leverages several quite sophisticated tools. Right, so the virtual agents decided first to use this large protein language model called ESM as a way to quantify and filter for potential nanobodies that have high stability and likely to be functional. Right, and then given this initial filtering by ESM, then the virtual agents decided to use AlphaFold multimer to basically to predict the structure, right, of this candidate nanobody together with the spike protein from the recent COVID variants. Right? So once you get this predicted structure, then they decided to actually feed this into Rosetta, which does more of like a physics-style molecular dynamics to relax that structure and compute the binding energy. Right? And then the virtual lab decided to actually do this 4 times iteratively to create like a good set of candidates. Right? While each of the individual tools like AlphaFold have been used before, right, in previous studies, I think the way that the virtual lab actually puts these different tools together into this workflow is quite interesting, and we haven't seen this in previous publications. And in addition, I think the virtual agents also made some interesting modifications and extensions of some of these tools. Right? For example, with this ESM, the protein language model, there are different ways you can actually use this to compute scores of different proteins. So the virtual lab actually created its own scoring function on top of the ESM model, which is different from what people have published before. And I think that's also quite an interesting innovation. Great. So all of this work is now done by the virtual lab with some sort of high level but relatively minimal guidance provided by us, the human researchers. And where the human feedback is important is on actually doing the real world experiments to help to ground the virtual lab designs and predictions. Right, so this is where we are fortunate to work closely with our collaborators, John Pak's group at the CC BioHub. And we have the virtual agents, they designed a set of nanobodies, and then we can actually then make these nanobodies physically and then test how well do they actually bind to the spike protein, right, of SARS-CoV-2 from the recent variants. So here are 2 structures, right, of 2 of these nanobodies designed by the virtual lab. And we basically tested these nanobodies against one of these recent variants. It's called the JN1 variant, which started to spread earlier this year. And we also tested them against the original Wuhan variant of the virus all the way from almost 5 years ago. And in the bottom here, it basically shows the real experiments from these affinity assays called ELISA assays. Right, so first, if we look at these red curves in the bottom, right, so the lowest red curve basically shows that if we start with the existing nanobodies that people have designed and used before, right, how well does it bind to this recent JN1 variant? And basically, there's no binding signal at all to the recent JN1 variant, right? And then after the virtual lab's optimization, right, now we actually, that's the solid red curve. Now we can actually see that there is some quite promising binding signals, right, to this recent variant. And then the blue curve also shows that in addition, right, these nanobodies designed by the virtual lab also binds strongly to the original Wuhan variant, right? It actually gained even some stronger binding affinity compared to the original nanobodies. And the reason why this is particularly exciting to us is that it's actually quite difficult, right, to have some nanobody that actually bind well across a very diverse spectrum of the virus. And that's why, like a vaccine or therapeutic that works well in 1 year may not work very well when the virus evolves and mutates. But here, the nanobodies designed by the virtual lab actually does seem to show some promising binding profiles across these 2 very diverse variants, right? From the one from 5 years ago to the one from earlier this year. I think that's very promising and exciting. And I want to be cautious here that I think we need to do a lot more additional experiments and optimization, right, to validate these designs. But I think there's already quite a nice demonstration of how the virtual lab is able to take a very open-ended research problem and then come up with its own creative computational workflows, making some innovations there, and then actually really designing something that's really useful and potentially quite impactful for the scientific community. So we are extremely excited about the virtual lab of these sort of teams of AI agents really being sort of the future of research and development going forward. Right? And we view this as sort of really a nice way for human researchers to then be able to collaborate with these virtual scientists that have diverse and complementary expertise. So I'll stop here, and then maybe we can have more discussions during the discussion period.
Thank you, James. That's a great summary. Appreciate that. So a lot of questions come to mind, James. The first question that comes to mind is that you use GPT-4o for these large language model agents. Can you comment a little bit on how you chose to use GPT-4o? And so this is a two-part question, because part of it is how did you select GPT-4o to start with? And also, what's your take on models such as GPT-o1, for instance, where OpenAI makes certain claims about the reasoning capabilities and things like that?
Yeah, so that's a good question. I think we also experiment with other language models. I mean, one of the nice things about the virtual lab is that it's quite flexible. So we can build our scientists on top of any large language models. So in particular, when the models improve over time, right, when they improve their reasoning as we expect them to, then our scientists actually gets better and better over time. Right? So for example, now we're actually also experimenting using the O1 model for some of these scientific discussions. And you can imagine that better reasoning and deeper reasoning also helps with better scientific discussions.
Fantastic. James, another question that comes to mind is when you were actually examining the content of the conversations that were happening between the agents, there is this notion of autocurricular learning. Did you see these models in agents in early iterations to have more rudimentary type conversations, and over time they got better and more sophisticated? Or were they pretty much just conversing quite readily right from the start because they were benefiting from the world model inside the underlying LLMs?
Yeah, it's a good question. So one of the nice things about the virtual lab is that all of their discussions happens in English, in natural language. So it's actually relatively easy for us to look at the summaries of their meeting logs and see what did they discuss, and so on. All right. So we do find some interesting cases where, for example, the agents actually disagree with each other. Right. So like their thinking actually evolves over time, because when they disagree, then they have to reach some sort of resolutions. And also having this critic agent often providing some more like constructive or negative feedback, right, also helps the agents to evolve and sharpen their reasoning over time. So in our case, we actually do have like a memory mechanism, right, whereby the agents actually, they have all of their previous meetings, and they have their external memory of what's been discussed happened before. So that does cause the agents to basically continues to learn from their previous meetings and previous experiences, and then adapt over time.
Fantastic. So James, you talked about the application of virtual labs to drug discovery. How do you see this kind of technology being transferred into other domains, such as, let's say, finance, or into other types of domains where there's lots of data and this kind of technology could benefit?
Yeah, so thank you. So we set up this virtual lab to be very flexible, right? So one part of our design decision is that we actually wanted the professor agents to train and to design their own students with the relevant expertise. And I think the power of that is that then it's relatively easy for us to adapt the virtual lab to other domains, right? So I could look at finance or education, right, or climate science or physics. And each of those cases, there will be different expertise that are required, right? But it's actually up to the professor agent then to be able to essentially teach the sub-agents, right, to develop the relevant expertise and to then apply this kind of structure to their relevant problems.
Great. James, another question that comes to mind is I recall last year we had discussed a paper that you were a co-author on, a very impactful paper about how ChatGPT's behavior changes over time. Now, given that now we're using large language models and we have these active software entities that are using the knowledge of those models to do things, how do we make sure that the behavior of the underlying models as it changes over time, that we can track the changing behaviors of these agents that are using those models?
Yeah, so there's a few things that we find to be quite helpful. One is that, as I mentioned, we actually do a lot of parallel meetings, right? I think that's basically one way for the agents to both explore a broader diversity of ideas, but also then to be, to reach some consensus decisions, which is more likely to be stable over time, right? This is sort of analogous to, like, if you ask me the same question on 2 different days, I might give you different answers, right? But if we look at across the aggregate of my answers, then that's something more likely to be stable over time. The other big thing is that, so here the agents are, and I think this is something that's perhaps more salient in these scientific applications, is that we want these agents to actually use these quite sophisticated tools in scientific domains, right? So here the agents actually learns to use tools like AlphaFold, right? These protein language models. And these tools, I think, also helps to improve the capability of the agents, but also to stabilize them, right? Because then if you're using this external tool and the tool doesn't change over time, right? Where this particular tool, AlphaFold, you can use a particular version that doesn't change over time, right? And I think that also makes the whole process, the computational workflows designed by the agent itself, to be quite stable and reproducible.
Okay, great. And James, the last question that comes to mind is then, what's the research horizons on this line of research? What's the next steps? Where's the future work? Where are you taking this research going forward?
Yeah, so we're extremely excited about this vision, right, of AI scientists that can become our partners and can help us and help our real scientists to improve and to speed up the innovation process, right? So I think the virtual lab is a good step in that direction, and there are a lot of exciting things we're building on top of the virtual lab that I think will really become, I hope for this to become sort of the future of R&D.
Thank you very much, James. As always, this was a very thoughtful and thought-provoking talk, as always. And before we wrap up, I would like to just make sure I invite you to come back again and talk to us about TextGrad, because you have so many high-impact papers and such phenomenal research that every time we talk about one topic, there's a new topic queued up. So would love to have you come back to the forum to talk about TextGrad and many other topics as well.
Yes. So I'll be delighted to come and talk about TextGrad, and also just say that all of these things are related under the hood, right? Because TextGrad is like a way to help us to optimize the agents systematically, right? And as we build the virtual lab with these multi-agent interactions and collaborations, then these kinds of TextGrad style optimization also becomes extremely useful.
Thank you so much for joining us today, James, and look forward to having you back very soon.
Thank you.